Hello guys, today we're going to cover the first part of hematology physiology series, and we're going to talk about mainly red blood cells. Let's start. Before we start, let's see what we're going to cover today. The first is blood composition in a very general way, and afterwards we'll go through RBCs, their suspension stability or the sedimentation rate, right? Hemoglobin derivatives, ion metabolism, erythropoiesis, and hemolysis. All right, so let's start. What is actually blood? It is technically considered as a connective tissue. And if we place blood in a centrifuge, it will separate into its component, right? As you can see here on the picture on the right. The densest part, right? The densest part, which is in the bottom of the tube, is comprised of erythrocytes, which actually form hematocrit. A hematocrit is also called a packed cell volume, and it is the volume percentage of red blood cells in blood, measured as part of a blood test, and normal hematocrit is 45%, right? 45% is a normal hematocrit. HCT or PCV, you can see different types of naming it. The least dense part is the yellowish plasma. You can see, as you can see, it comprises 55% of the blood. And these two parts, big parts, are separated by buffy coat. It is a white layer containing platelets as well as leukocytes. Well, what is the function of blood? Blood is responsible for carrying oxygen, nutrients, hormones, waste products, right? Like carbon dioxide, for example. It serves as well to regulate pH in various tissues. It maintains body temperature and prevents infections. In order to understand those functions properly, we need to go deeper into each component of the blood, but we won't go very deeply into some of them. We'll go deeply into red blood cells mainly. But first, let's go to plasma. Blood plasma is a fluid which contains 90% of water and 10% of solids. And those solids are ions, proteins, hormones, minerals, glucose, right? Carbon dioxide, gases, electrolytes, antibodies, antigens. We said that blood serves as, has a very huge role in immune system, right? So blood plasma actually carries those antibodies and antigens, exogenous substances such as drugs and microorganisms, for example, right? The most abundant plasma protein is called albumin. And since it's so abundant, it's like 60% of all proteins, it significantly contributes to the osmotic pressure of the blood. Like osmotic pressure of the blood, it draw water from tissues into the bloodstream. Blood pressure forces blood out of the capillaries. So if protein is low, it would lead to edema. The part of osmotic pressure that make up proteins is called oncotic pressure, right? So I meant oncotic pressure. Albumin is followed by a variety of globulins, and there are 35% of them out of all proteins. There are different types: alpha, beta, gamma globulins. If we talk about alpha and beta, they transport fats because fats are not water soluble, so they need some transporter. And these proteins which transport fats are called lipoproteins. Then we have transferrin. Transferrin is beta globulin which transports iron. Then we have, for example, ceruloplasmin. Ceruloplasmin is an alpha globulin which transports copper, but actually it's not the only function of ceruloplasmin, and we'll talk about it more in a bit. For example, immunoglobulins as well. Now they play a role in the immune system, right? The next type of proteins are blood clotting proteins, and they're only 5%. These are mainly prothrombin and fibrinogen. And if you put plasma in a glass beaker, it will form a clot on the top, right? These are all the clotting factors, including fibrinogen. So the blood plasma without clotting factors is called blood serum. So if you remove that clot, then you have serum. In addition to proteins, what else is in plasma? You actually can see here, stop the video and read and go through all these molecules that are in plasma. Minerals, for example, sodium, potassium, they make up osmotic pressure. Some of them, they have a huge role in pH, etc., etc. You just go through them and probably just Google some parts or read from your textbooks about some parts. It's a huge topic to cover. But what we're going to talk about is red blood cells. So the other part of the blood, we said first part is plasma, and the other part is blood cells. We had hematocrit, right, the densest part, and in between we had buffy coat. Buffy coat contained leukocytes and thrombocytes. Leukocytes, or white blood cells, are much less common than red blood cells, and their role is also very different from that of red blood cells. They're primarily involved in immune responses, recognizing and neutralizing invaders such as bacteria and viruses. We can divide white blood cells into two groups, granulocytes and agranulocytes. Regarding granulocytes, we have neutrophils, eosinophils, and basophils, all of which have granules in their cytoplasm when stained and viewed on a microscope, for example, for sure. The other group is agranulocytes, and it includes monocytes and lymphocytes, which do not have granules in the cytoplasm that we can see. All right, then let's go to platelets. Platelets, also called thrombocytes, are cell fragments involved in blood clotting. Again, platelets are not cells, right? They are made up of, they're kind of parts of a huge cell called megakaryocyte, but it is not a cell, all right? It's just, that's why it's called platelets, and it is involved in blood clotting. But it is not the topic of our today's lecture. That's the main topic of today's lecture is RBCs. Well, red blood cells, or erythrocytes, are specialized cells that circulate throughout the body and deliver oxygen to tissues, right? From the lungs to the tissues and carbon dioxide back to the lungs. In humans, red blood cells are small and biconcave. Biconcave means that they have the thinnest part is in the center. And red blood cells do not contain mitochondria or a nucleus when mature. Why is it so? Well, these characteristics allow red blood cells to effectively perform their task of oxygen transport. Well, small size and biconcave shape increase the surface area to volume ratio, improving gas exchange, while, for example, lack of nucleus makes additional space for hemoglobin, a key protein used in oxygen transport, and we're going to talk about it in a bit. Lack of mitochondria keeps red blood cells from using any of the oxygen they're carrying, maximizing the amount delivered to tissues of the body. In the lungs, red blood cells take up oxygen, and as they circulate through the rest of the body, they release the oxygen to the surrounding tissue. So they need to have this this opportunity to release, right, in the tissues and to take up the oxygen in the lungs. But as well as actually red blood cells also play an important role in transport of carbon dioxide. Carbon dioxide is a waste product and it travels from the tissues back to the lungs, right? Red blood cells have an average lifespan of 120 days. Old or damaged red blood cells are broken down in the liver and spleen, mainly in spleen, and new ones are produced in the bone marrow. Red blood cell production is controlled by the hormone erythropoietin, which is released by the kidneys in response to low oxygen levels. And we're going to talk about it in a bit deeper way. And actually, this negative feedback loop ensures that number of red blood cells in the body remains relatively constant over time. All right, so erythropoietin is released to in response to low oxygen level. This is what you need to recall and to remember. All right, actually, when in circulation, the red cells remain uniformly suspended in plasma. If we take a specimen of blood and add an anticoagulant there, and is stand in a glass tube, usually it's used like very thin glass tube, the corpuscles, being heavier than plasma, are found to sediment, right? Sediment gradually at the bottom of the tube. While the plasma remains as a clear supernatant fluid, as you can see over here, right? This is the plasma and this is the corpuscles, right? This process is called sedimentation, and the rate of settling down, or like the rate of sedimentation, is known as erythrocyte sedimentation rate, ESR. Erythrocyte sedimentation rate is measured as the length of a clear column of plasma after 1 hour, right? So if you can see here, normal values of ESR in males and in females, it is in millimeters per hour, right? Because like it's the length of a clear column of plasma after 1 hour. So here you can see it. The sedimentation mechanism involves 3 stages, first stage is called aggregation. It's the initial stage in which piling up of RBCs takes place. The phenomenon is known as rouleau formation, and you're gonna meet this term, this terminology more often. Then we have a stage of sedimentation. It's the stage of actual falling of RBCs. And the last one is packing, stage of packing. This is the final stage and is also known as a stationary phase. Well, in this, there is a slower rate of falling during which packing of sedimented RBCs in column occur due to overcrowding, right? Why actually do we need ESR? The higher the ESR, the lower suspension stability of the red blood cells, right? ESR actually depends on elements in plasma. And it is a widely used test, blood test, but it is not specific for any disease. But it is widely, again, used in combination with other tests to determine the presence of increased inflammatory activity. Why so? Because it particularly increases in fibrinogen, immunoglobulins, and C-reactive protein, right? So if we have any kind of inflammatory state, like if we have infectious condition or inflammatory or degenerative or malignant conditions that are for sure associated with changes in plasma proteins that I've said already, fibrinogen, immunoglobulins, and C-reactive proteins, it leads to the increase of sedimentation rate. Actually, ESR is also affected by many other factors, including anemia, pregnancy, different pathologies and drugs. And you can again stop the video and read and go through all those causes of significantly raised ESR or reduced ESR. But it is not what you need to know right now during your physiology, but you will need it in pathology. Okay, let's talk about hemoglobin. What is hemoglobin? Hemoglobin actually is a very crucial part of our RBCs. It's the molecule, the protein that it's actually a metalloprotein that binds oxygen, but not only oxygen, and we'll talk about it. Hemoglobin contains heme and globin. Globin chains are large globular proteins, right? And there are several different kinds of them, as you can see, alpha, beta, gamma, and delta. And those are used to make different molecules found in our RBCs. All hemoglobin molecules are made up of 4 chains in 2 pairs, right? So please keep it in mind that all globin chains are, all hemoglobin molecules are made up of 4 globin chains in 2 pairs, all right? The other component of hemoglobin is heme. It is a molecule which is not peptide. It's actually, you can see the structure over here. It's made up of nitrogens and hormones and carbons with an ion in the center. Actually, the ring around the ion is called a porphyrin ring. And iron in ferrous state, Fe2 plus state, is what is going to bind oxygen. There are three major types of hemoglobin found in the human body. Actually, there are more, and even in human body, but we're gonna talk about after birth, like a hemoglobin found after birth, all right? And like major types. First, the adult type. The most common type found in adults is hemoglobin A. It's 95% of all hemoglobin, and it has two, recall I told you that all hemoglobin molecules are made up of 4 chains in 2 pairs. So here we have 2 alpha chains and 2 beta chains, right? So in general, 4 chains in 2 pairs. The other hemoglobin is called A2, where sometimes you can see hemoglobin B. It's adult type as well, and it's less common type, but it is still physiological type, and it's made up of 2 alpha and 2 delta chains. The last but not least is hemoglobin F. Hemoglobin F is a fetal type, and it is made up of 2 alpha and 2 gamma chains, and it is involved in transportation of oxygen from the mother's bloodstream to organs and tissues in the fetus, right, through placenta. So it means that fetus doesn't breathe in oxygen, but it takes up from the bloodstream, from placenta, right? So that's why hemoglobin has a different structure from the adult type, and it leads to the fact that it binds oxygen more strongly. The levels of hemoglobin F actually remain high even after birth until the baby is roughly 2 to 4 months old, right? The last 2 types I'd like to talk about are seen in pathological conditions. First is seen in carbon monoxide poisoning. Well, carbon monoxide is gas and it binds to iron in heme with 240 times affinity to oxygen. When it does so, it forms a structure called carboxyhemoglobin. And because it binds so avidly to hemoglobin, it blocks binding sites for oxygen, and therefore less oxygen can be absorbed by hemoglobin. All right. This creates what's called functional anemia. Even though there is enough hemoglobin, enough red blood cells, the body behaves as if it was anemic. Actually, it also blocks other binding sites from offloading the oxygen. It means that oxygen remains bound to hemoglobin. Recall that I said that in tissues, hemoglobin needs to release oxygen through 2BP pathway. It's more about biochemistry, but it does so. But due to carbon monoxide, actually CO is an allosteric modifier of hemoglobin, which leads to the fact that oxygen remains bound to hemoglobin even in the tissues. Strongly binds to hemoglobin. Second pathological condition is called methemoglobinemia. So most iron in hemoglobin is normally in a reduced state, right? Which is a ferrous state, Fe2 plus state. But very small amount, normally very small amount is found in Fe3 plus state. And this hemoglobin is called methhemoglobin. Actually, this hemoglobin is bad because it cannot bind the oxygen, and some pathological conditions can cause the excess of methemoglobin, and this can lead to hypoxia, low oxygen levels in the blood. For example, there is a number of drugs that can cause it. As I've told you, one of the main part of hemoglobin is iron. It's, if you recall, it's over here in heme, in the center of heme, and iron is what actually binds oxygen. Iron is actually the most essential trace element, I'd say. Total body iron content is 3 to 5 grams. And our daily dietary requirement of iron is between 10 to 20 milligrams. It means that we need to take up 10 to 20 milligrams per day. Women actually require a bit more of iron due to physiological loss of it during menstruation. 75% of iron is present in blood. The rest is in liver, bone marrow, and muscles, as you can see here. Heme is the most predominant iron-containing substance. That's why it's like 75% in blood. Let's talk about absorption. Actually, iron is absorbed from upper small intestine by divalent metal transporter 1, DMT1, as you can see here. It is located on the apical surface of the apical surface of enterocytes, and the observed state of iron is ferrous state, Fe2+. We receive Fe3+ state of iron, and upon reduction to Fe2+ state, ferrous form is absorbed. Why so? It's because it's more soluble, so it's more readily absorbed than Fe3+. The iron entering the mucosal cells by absorption is oxidized again to ferric form, Fe3+, and by the enzyme ferroxidase, and actually it can form ferritin by the combination with apoferritin, and ferritin is a temporary storage form of iron in mucosal cells and enterocytes. From mucosal cells, iron may enter the bloodstream. Actually, we've got the other type of iron intake, I'd say. It's splenic recyclation, right? So if we have spleen and we have a iron salvage pathway. That means that from the spleen, old erythrocytes that are broken down in spleen go back to to our body and serve further. So splenic reticular endothelial macrophages actually recycle iron, right? From senescent red blood cells, as I said, and from both types of cells, from splenic cells and from the enterocytes. Iron is released via ferroportin with aid of hephaestin. What it means, for example, look at this. Ferroportin is a transport protein that leads to the exflux of Fe2+ state. But then we can use hephaestin, right, in order to oxidize Fe2 plus state to Fe3 plus state. So iron in the end enters plasma in ferrous state, Fe2 plus, right? Fe3 plus, sorry. It can also be oxidized to ferric form from by a copper containing protein, right? Ceruloplasmin, I told you before about it. It's a type of globulin, and it has peroxidase activity. Hepatic hormone hepcidin, which you can see here, regulates the afflux of iron from these cells by regulating the stability of ferroportin. Look, as shown here. The synthesis and secretion of hepcidin by hepatocytes are influenced by iron level in the body, as well as conditions that affect iron metabolism indirectly, such as inflammation, erythropoiesis, hypoxia, etc. In plasma, ferric iron binds with a specific iron-binding protein called transferrin. So, transport form of iron is transferrin, right? It is a glycoprotein synthesized in liver. And one molecule of transferrin can transport 2 ferric atoms, and the half-life of transferrin is 7 to 10 days. Why do we need to know that? Actually, in different types of anemia, we need to measure iron level and total iron binding capacity. Actually, in iron deficiency anemia, serum iron level is decreased and total iron binding capacity is increased. What is it? Now, total iron binding capacity measures the total amount of iron that can be bound by protein in the blood, right? And since transferrin is the main protein that transfers iron, so we need to know its amount. And actually, this TIBC, total iron binding capacity, can indirectly show us the amount of transferrin. Okay. Iron is stored in liver, spleen, and bone marrow, as I've said before, in the form of ferritin. Ferritin is an iron storage protein. It stores iron in ferric form and actually releases it through channels in a controlled fashion. Ferritin levels in blood is an index of body iron stores. Actually, it's an acute phase reactant protein and is elevated in inflammatory diseases. There is another iron storage protein, which is called hemosiderin, and it accumulates when iron levels are increased. Regarding excretion, the normal iron excretion is about 1 to 2 milligrams per day. The major excretory pathway is through intestine. Actually, iron is not excreted in urine, but in some conditions, such as nephrotic syndrome, loss of transferrin may lead to increased loss of iron in urine. Okay. Let's go to erythropoiesis. Let's talk about hematopoiesis in general. Hematopoiesis describes the production of cells that regulate in the bloodstream, right? Erythropoiesis is the process by which red blood cells are produced. On average, the body produces like really a lot of red blood cells per day. And erythrocytes arise from a complex line of cells, and you can see it here, right? This is the hematopoiesis in general, and those lines are very strictly regulated. The site of erythropoiesis changes throughout life. In the very early fetus, it occurs in the yolk sac. From 2 to 5 months gestation, it occurs in the liver and spleen before finally establishing in the bone marrow from about 5 months gestation. Actually, in children, erythropoiesis can occur in the bone marrow of most bones. However, in adults, it only occurs in the bone marrow of the vertebra, sternum, ribs, femur, and several other bones. When erythropoiesis is inadequate in the bone marrow, this can trigger extramedullary hematopoiesis. It means that hematopoiesis occurs outside the marrow, and it is commonly seen in different types of thalassemia, for example. Let's go to the stages of erythropoiesis. The production of all blood cells begins with the hemocytoblast. Right, here it is. It is a multipotent hematopoietic stem cell. Actually, hemocytoblasts have great power of self-renewal of any adult cell. They're found in the bone marrow mainly, and actually can be mobilized into circulating blood when needed. Some hemocytoblasts differentiate into common myeloid progenitor cells, as you can see over here on the left, which go on to produce erythrocytes, right? And actually some other types of cells as well. The process of erythropoiesis itself involves several stages. First, they become normoblasts or erythroblasts, which are seen here. They still contain some organelles and nucleus. If you recall, I told you that red blood cells do not contain mitochondria and nucleus. So afterwards, reticulocytes are made, and reticulocytes can be thought of as immature red blood cells. And in some types of anemias, it is important to see reticulocyte count. Finally, reticulocytes in several days actually lose their remaining organelles, and they mature into erythrocytes, right, as you can see here. And erythrocytes are fully mature red blood cells, and they can normally survive for around 120 days, as I told you before. Erythropoiesis is driven mainly by the hormone called erythropoietin, which is a glycoprotein cytokine. It is secreted by the kidney, as I've told you before, and it is constantly secreted at a low level, sufficient for the normal regulation of the erythropoiesis. However, in the erythrocyte level, it becomes inadequate, the blood becomes relatively hypoxic. And as I've told you before, when the blood is hypoxic, which means that there's a reduced partial pressure of oxygen, mainly, and such kind of blood goes to the kidneys. This is detected by renal interstitial peritubular cells that start EPO production, right? And EPO acts in the bone marrow to stimulate increased red blood cell production. So it actually causes hemoglobin levels to increase, subsequently causing the partial pressure of oxygen to rise, and therefore the EPO levels, erythropoietin levels, fall. So this is a negative feedback loop by which erythropoiesis is regulated. Let's go to the last part, which is hemolysis. Hemolysis is the destruction of red blood cells. As I told you before, red blood cells live approximately 120 days. During this time, they undergo various metabolic and chemical changes, which result in a loss of deformability. Under normal circumstances, macrophages of the mononuclear phagocyte system or reticular endothelial system, they recognize these changes and phagocytize the aged erythrocytes. The organs that are doing so are spleen and liver mainly. The macrophages in the spleen actually are especially sensitive to RBC changes, whereas the macrophages of liver, they're called Kupffer cells, if you recall, they detect and destroy more severely damaged RBCs. So the main hemolytic process actually normally is seen in the spleen. Actually, there are cords of bilirubin, as you can see here, and on the left you can see them. These are vascular channels that end blindly, and actually normal erythrocytes that are not damaged, they are not old, young and very healthy erythrocytes, then they can go through those cords of bilirubin. The damaged and old erythrocytes are not so flexible, so that's why they cannot go through those cords of bilirubin, and they remain there. We can say that hemolysis can be extravascular and intravascular. Extravascular hemolysis is seen in spleen and liver, as I've told you, and intravascular hemolysis is not a good state. It's not physiologically normally, and it shouldn't be seen. If we see here, as I've told you, the majority of RBC degradation occurs extravascularly. A hemoglobin, as a part of RBCs, is hydrolyzed into heme and globin. Globin is further degraded into amino acids that return to the amino acid pool, and the ion from the heme returns to the plasma via ferroportin, as we covered before. The remaining protoporphyrin is degraded through a series of biochemical reactions in different tissues and organs. While protoporphyrin is inside the macrophage, heme oxygenase acts on it, breaking the porphyrin ring to yield a linear molecule, biliverdin. Right, here you can see. Biliverdin actually is green, and then it is reduced to bilirubin, which is actually yellow molecule, and it is nonpolar. And nonpolar yellow bilirubin is secreted into the plasma. Since it is hydrophobic, this bilirubin must bind to albumin to be transported in plasma to the liver. And actually, bilirubin albumin complex is called non-conjugated complex, enters the liver, and then bilirubin dissociates from the albumin and is joined with two molecules to form bilirubin dichloroanide 2. And afterwards it go to and actually the like two molecules are sugars, sugar acid molecules that make the molecule polar and water soluble. This is what we need for bilirubin to be excreted. It is called conjugated bilirubin. Bilirubin that is released from macrophages is called unconjugated, and after the liver, it becomes conjugated. Conjugated bilirubin is excreted as bile acid into the intestines. It is oxidized by gut bacteria into various water-soluble compounds, collectively called urobilinogen. Most urobilinogen is oxidized further to stercobilin and similar compounds that give the brown color to stool, which is the ultimate route of for secretion for protoporphyrin. But since it is water soluble, some conjugated bilirubin and urobilinogen are absorbed from the intestines and can be detected in the plasma. Then they go to the kidneys and are excreted by kidneys, so which gives the urine its yellow color. So this is in general how hemolysis occurs in physiology, but it is very important for you to understand different stages of physiological normal hemolysis in order to understand what happens, what clinical conditions what lab findings we can see during different pathological states. For example, we have like you can see here, hemolysis is seen in pathology normally, and it causes normocytic anemia. Normocytic means that mean corpuscular volume of the red blood cells is normal, with a normal range. And this anemia, during this anemia, you can see increased elevated unconjugated bilirubin, right? Why is it so? Because the capacity of liver is exhausted. Liver is exhausted, so it cannot convert all bilirubin to conjugated form. So we've got elevated unconjugated bilirubin. Then we have increased reticulocyte count because bone marrow reacts properly. And the last but not the least, we have elevated plasma LDH. Elevated plasma LDH is actually, LDH is lactate dehydrogenase, and erythrocytes, as I've told you before, they do not have mitochondria. So they use anaerobic glycolysis. And in anaerobic glycolysis, LDH actually converts pyruvate into lactate. So for now, I would say that's it. Thank you very much for your attention. If you have any questions or any notes, or if you mentioned any mistakes, please feel free to comment below. Thank you very much and have a nice day.